All right, I think we've got everyone back from the break. So we're going to start off. Andy gave us some nice motivation for the whole theme of the day, which is calibration and validation of those peptide assays that we started building yesterday. So we kind of ended yesterday with the idea that we had some sort of hypothesis. So if you got a chance to get to that plasma tutorial, that's great. And if you didn't, the whole idea with the plasma tutorial was if your boss, or for some reason you, came up with some set of 6 proteins, including a lot of the apolipoproteins, how would you go about building a proteomics assay for those 6 proteins? There's a lot of places you might start. You might start with absolutely no idea what peptides you'd want to look for. So maybe you'd set up Skyline to just do what's called an in silico digestion. You would tell Skyline, take this protein sequence and give me every single possible peptide I might get from that protein. You might imagine that adds up to a lot of peptides very quickly, especially when you start considering like one miscleavage or potential modifications. So maybe an easier place to start would be with some prior knowledge. So with Sue, we talked about using things like spectral libraries. So that might give us, based on shotgun data, on data-dependent acquisition, what peptides can I even see by mass spectrometry? What peptides fly, what peptides digest? So we might go from the space of every possible theoretical peptide that exists for a protein to things that we have observed in a mass spectrometer. And then the last phase of that tutorial yesterday showed you, well, what if you had a protein standard for each of those 6 proteins, and you analyze the protein standard? So now not only do you have every possible peptide that might exist, just the peptides that have a single MS-MS spectrum, because DDA will just give you that one single MS-MS spectrum, the best one. What if now you had actual chromatograms for all of your peptides, and you could start doing some of these calibration and validation experiments to decide the best peptide? So as you get more and more prior knowledge, you're getting better and better candidate peptides that will act as good proxies for the protein that they're supposed to be representing. So we kind of got through this whole workflow yesterday, and if you need kind of some review material to synthesize it, we put out a review a couple of years ago going over the entire Skyline ecosystem. So it might be a good reference point to help put into context some of these things. With this short lecture this morning, I really only have 2 objectives for you to walk away with. One is I want you to be thinking about these validation criteria that we'll talk about and which ones you might need to use in your own experimental needs and how you might modify these approaches for whatever your matrix of interest is, whatever your hypothesis is. I know a lot of people are doing interesting modifications. And then I want you to think about the requirements for quantification, like what does it mean to detect something and what does it mean to quantify something by mass spectrometry? So we're first going to go over kind of what even is a figure of merit. This is something analytical chemists we like to talk about a lot, but really it's just kind of our own jargon. Then we're going to talk about different approaches and guidelines. Then it's a little bit, everybody has their own set of guidelines that they like to follow. And then finally, I'm just going to highlight one of those guidelines, which Sue talked about yesterday, which is the CPTAC, Assay Portal Recommended Validations. So just to review one more time, yesterday we were talking about how do we pick the targets, which would be the precursors, and the transitions, which are those product ions after we fragment the peptides, to build the target list that sits on that left-hand side, usually, of your Skyline document. We talked about collision energy to optimize how much product ions that we could get. We talked about retention time, that liquid chromatography system that we like to ignore in LCMS, and how important the liquid chromatography can be in our method development. And then I had given you some slides that talk about modifying your liquid chromatography gradient for separating versus washing. And finally, I had some information about the concept of trapping or a pre-analytical column. But kind of by this point in the course / in your experimental development, You've got this assay. You've built this assay potentially from scratch or with spectral libraries. You maybe ran a couple acquisitions to test your method, maybe your scheduling, and you had to adjust how wide your windows were going to be so that you could balance how many targets versus how long your method is. You evaluated those chromatograms. You removed any interferences. You built an IRT calculator. And then you just kind of keep doing this, refining your method, refining your method. This is where it starts to become the step of thinking about calibration and validation. So Andy gave us good motivation this morning, but I want to motivate you a different way. If all I told you was the chromatogram on the left and the chromatogram on the right are the same peptide in two different samples, could we say this sample has more abundant peptide than the other? Certainly the peak area is more intense. We have a higher peak area in this sample on the left, and we have a smaller peak area on the sample on the right. Without doing anything else, we might say, sure, this peptide is more abundant, so therefore I would assume that the protein is more abundant, that this peptide came from, the protein's more abundant. But we can't actually say that because mass spectrometry signal is not inherently calibrated. What if I told you this was ran today and then this sample was ran 20 days later? What if I told you this was on a Q exactive that everybody loves because it's super clean and has great intensity, and this is on the exact same Q exactive sitting next to it, and it's the same sample, but this Q exactive is a little bit dirtier and nobody likes to use it, so nobody signs up for it. We can't tell what reason caused these two peak areas to be different unless we've done calibration and validation. So things like using an internal standard or using an external standard, some sort of single point, multi-point, external, internal calibration to validate that those two signals are actually different in the sample. And that's kind of the whole theme of today. Now you've got these targets and you want to trust that the quantities that you're reporting are real. So Andy told us a little bit about calibration curves. And just in case you're not an analytical chemist, the idea behind a calibration curve is just that you have some set of samples, some dilution of samples in the matrix that you're interested in. So Andy talked a lot about plasmas. So they have specific plasmas you can get that are like depleted of your protein of interest. That might be one way. But I think Andy's lab lately has been using an equivalent organism that is evolutionarily diverged enough that you won't have overlap is one way you can do a matrix like that. A lot of times this is with some sort of synthetic isotope that is spiked in at different concentrations from a very, very high concentration all the way down to a blank that has none of the peptide target you're interested in. And you would expect, as the concentration decreases, that your signal off your mass spectrometer should also decrease. And the range when this signal is the 1-to-1 is what Andy was saying is the linear range, where that slope would be 1-to-1.
Sorry, this might be a silly question, but
no such thing.
when you're adding these different concentrations, are they all running the same run, or?
That is a great question. Are all of these concentrations run on the same run, or how do you acquire these? So let's imagine we have 10 samples ranging from very high amount of analyte to the blank. When you run these on your mass spectrometer, you run it from the blank all the way up to the highest concentration point, not randomizing. So as opposed to like a biological experiment where you'd want to randomize everything and block it, for a calibration curve, the blank up to the highest point, and then maybe a wash to make sure you have no column carryover from that high point. And then again, the blank up to the highest point, another wash or whatever, and however many replicates you're going to do. So that's actually a really good question, because if you were to do this the opposite way and do the highest point down to the blank, you would potentially have carryover, which is, carryover just means there's some sample that's still stuck on your column that didn't come off. Depending on how you set up your LC method and your peptides of interest, this can be worse or not so bad. So if you run from the blank up to the highest, you don't necessarily have to worry too much about that. Another question?
Yeah. When you're talking about the kind of how much or how low of one particular peptide I can quantify with my, but you do not compare range across several peptides, right?
Each one of these curves is for one peptide, one analyte. And even I know Sue in the past has done each individual transition for each, because different transitions, even different product ions, might have different calibration curves depending on if they're interfered with or not. So I think most commonly I see this at the peptide level. Sue is really hardcore and does transition level. The linear range, just to point out a couple more things like Andy was talking about, the percent coefficient of variation is if I did one of these concentrations and analyzed it multiple times, how reproducible is the peak area I would get. And there's a lot of different ways you can assess this, and we'll talk a little bit more about that in some of the later slides. But one way to assess this is to just straight up leave that sample in your autosampler. How stable is the peptide if it's just sitting in your autosampler for 4 hours, for 12 hours over the weekend because you're a lazy grad student, you don't want to go in. If it's degrading, your peptide's degrading, the variance will increase, so the CV, the percent CV, will increase. And a lot of kind of standard rules of thumb is a 20% CV is your threshold. Anything over a 20% is maybe too irreproducible. Mm-hmm. Matrix is extremely important, is super, super important. The matrix must be as similar to your sample matrix as possible. Identical would be best. So if you're using some types of calibration curve, you would have endogenous in here too. Your light would be in here as well. So there's ways you can do like reverse. Oh, Skyline automatically separates heavy from light. Oh yeah, we're doing all calibration curves all day in Skyline. I hope you're excited. I'm excited. I love calibration curves. So the reason the dynamic range kind of cuts off right here is because it's not linear. Your response off of your mass spectrometer is not decreasing forever. At some point, the signal on your mass spectrometer kind of levels out because you've hit a floor. And any lower input you analyze, even though you're decreasing the quantity of the analyte in your sample, you're not getting any change in the signal because you've hit the limit of that peptide in that matrix on that system. That point at which you kind of hit that limit is the limit of quantitation. So this limit of quantitation is an example of a figure of merit. Doing the percent CV and the reproducibility can be a figure of merit. These are peptide dependent in the matrix you're analyzing on the system you're using. So if you took the exact same sample and exact same calibration curve and did it on one system, like a triple quad versus an old ion trap or something, you would get different figures of merit because you've changed the analytical platform. If you took the same peptide and put it in, for example, like an E. coli background, an E. coli lysate, versus a human lysate, like a human HeLa lysate, you would get a different limit of quantitation because you've got a different matrix now. You've got a simpler matrix versus a more complex matrix. And that's why it's so important that the matrix you make your calibration curve in matches as close as possible. Sue's going to talk tons about how to do this and what standards to use, but I just wanted to introduce these concepts because we're going to look at some of this data next. So why do we care about figuring out which peptide calibration curves are the best for our proteins of interest? This is a very busy figure that I'm going to walk you through. So I do all of my work in yeast because it's very easy and I like easy projects. So this is one protein in yeast that is extremely abundant. It's like the second most abundant protein in yeast. I've drawn its sequence as though it were just a line. This black line here. It's a membrane-bound protein, so there's some transmembrane domains that I've marked in gray. And then each of these colored boxes is a different peptide I detect by mass spectrometry. So I detect a lot of proteins. This is pretty decent coverage. You notice we don't detect every part of the sequence on this protein. There's some regions in a protein there's just no good tryptic peptides. Some regions have a tryptic peptide, but for whatever reason, they either don't digest or they don't fly. They don't ionize well is what we mean when we say they don't fly. For the regions I do have good coverage, the boxes are different colors because I've colored the boxes by their limit of quantitation. So the lower the limit of quantitation, the better. The lower the limit is, the smaller amount of peptide you can detect and find a change in the signal for a change in the quantity. So the lower the limit of quant, the lower that inflection point. And you'll notice there's really kind of maybe no rhyme or reason which peptides have a good limit of quantitation and which ones don't. A lot of these peptides show no change in signal when I've deliberately changed their quantitation. So it's something I detected, but I certainly did not quantify in the sense of change in signal, change in quantity.
Is the detection correlated with the size of the peptide?
So generally, rule of thumb, kind of a smaller peptide is supposed to fly better. I didn't show you the sequences here, but I do have the sequences up at the top for 3 of the peptides, where I have a really low limit of quantitation. This is a peptide where the change in quantity, the change in signal reflects a change in quantity for a very long range of concentrations. So this peptide looks fairly, I guess, normal, I would say, as far as a target you might pick for mass spectrometry proteomics. The peptide in the middle that has a mid LOQ is maybe a little bit long for what I might have chosen for this particular protein. But this peptide on the end, it does have a methionine, so maybe I wouldn't have picked it for quantification. But I really have not determined any obvious reason why some peptides have a lower LOQ than others. But this is just to motivate you. If I were to pick one peptide for this protein, depending on which peptide I chose, I would have an easier or harder time quantifying things over a broader range. And that's really the takeaway message I'd like you to get from this figure is doing the calibration curves, I can choose this peptide is the best proxy, quantitative proxy for this protein. Question over here?
Say there's no LOQs, does that actually always mean that there's just a very high CV for every membrane?
Yeah, for this particular example, it's just that the CV is very, very high and never meets that 20% CV criteria that we talked about before. But a lot of these peptides, it just looks like I took a shotgun and blasted the plot. There's no correlation whatsoever. Question in the back and then in the front?
So one question is if you have multiple times, like 50 times,
Mm-hmm.
you do calibration for each of the peptides. And another part is like if you have multiple peptides from one protein, do you represent just one peptide? Or
So each curve, each plot will be one peptide. You can mix all of those peptides into one calibration curve dilution set. So I think Sue's going to talk way more about this later, actually setting these samples up. This is just to help emphasize why we're going to bother with doing calibration and validation. Oh, one more, sorry.
Let's assume I have this 100 peptides. I don't know the exact quantities. I don't have the money to it. But
Yeah.
I have them all crude. If I want to do relative quantification across several samples and I just want to know what kind of intensity is reliable and what isn't, I don't need to know, right?
No.
I can just have on the x-axis x, 2x, 4x, and so on.
Yep, you could just do like a fractional dilution and just say like my highest point is 100%, and then I've fractionally diluted it down to 0% of my crude in the sample. But Sue's totally got you covered. I think we have a whole lecture just on calibration curves because they're that kind of difficult to grasp. Was there any more just on motivating you why we should care about picking the right peptide proxy for a protein? One more, yeah.
Is this based on digestion that the peptides are
It could be part digestion. This protein was exposed to trypsin. All these peptides came from the same digest. I just took a whole yeast cell lysate, digested it, and this is the different protein, the different peptide responses from this protein as it got digested.
So you didn't take just this protein and digest it?
Nope, the whole lysate. Yep.
Then probably this is because that peptide was similar to other, came from other proteins.
It might be with yeast. We don't have quite as bad a problem with isoforms. There's not a lot of splicing in yeast or anything, so homologous proteins might have peptides that are shared. But it's a combination. It sets me up perfectly. Why are some peptides have a lower LOQ than others? And it could be anything. So Andy kind of brought this up earlier where we're interested in the amount of protein in a sample, but we do all this downstream stuff to the sample, and what we measure is just a proxy for what existed in the original sample. So we talked about how are we denaturing. For me, a big problem in yeast is how am I lysing, because yeast have a cell wall. So my lysis affects how much protein I get in each sample. The denaturation affects how much protein unfolding. The digestion conditions we just saw can vary widely between different samples. We're separating peptides and almost inevitably losing a bunch of peptide to the column. We're trying to get them to fly. These are like fairly large molecules that we're ionizing, right? And then we're trying to measure them by mass spectrometry. We have to detect the MS2, right? And then not only detect one of those MS2, but multiple MS2 to get the points across the peak in the chromatogram. So we're actually manipulating the samples a lot. So the readout we get in the end is a peptide extracted ion chromatogram, but what we started with was like a whole yeast cell or a whole plasma aliquot. So even if we were to take some of those things out, so now let's imagine I have a mixture of equimolar peptides. So digestion isn't a problem anymore. Now we're just looking at putting it onto the mass spectrometer. So we're doing LCMS peptides from the same protein. I have an equimolar mixture of peptides from this one protein, and their range of response in the mass spectrometer varies massively. This is a log scale, right? So the range of peptide responses just in the mass spectrometer can be huge. So even if we were to control for lysis and denaturation and digestion, different peptides are gonna fly better or worse. So what do you do then to pick which peptide is gonna be the best proxy for a protein? And the answer is it depends who you ask. So there's a lot of guidelines, and it's kind of whatever fits your experiment the best. So Andy has a set of guidelines with Russ Grant and a paper I think most of the field just calls Lost Paradise Found paper. And then there's, this is kind of like a CPTAC, or not a CPTAC, molecular cellular proteomics journal, basically. Guidelines are in the fit for purpose paper. So we call this one Paradise Found, and this one is just called fit for purpose, basically. You'll notice the same themes. I'm going to show you some of these guidelines. So some of the guidelines from that fit for purpose paper say it depends what you're doing. So if you're doing like clinical proteomics, your calibration validation standards are probably going to be a lot more strict than if you're just some academic doing yeast biology. I probably don't have to be quite so strict in my calibration and validation. So it's kind of the fit for purpose idea of depending on what you're using your assay for, you might have different standards for your calibration and validation. Andy has put forth a set of guidelines, especially for clinical practice. And these are some of the data sets that we're going to be looking at after this lecture, which is supposed to be done in 5 minutes. Good to know. So we're going to look at that vitamin D binding paper that we heard a little bit about. And we're going to look at these different validation data sets in Skyline, so you can kind of see what does this data look like. We're talking a little bit now about how it might be collected, but what does it even look like? But the one I'm going to go into a little bit more detail with is the CPTAC assay portal. So CPTAC we heard a little bit about from Sue, which is kind of, can we even use mass spectrometry and proteomics in the clinic for cancer? They have a set of experiments that are proposed in their suggested guidelines. The first one is the response curve, which I'm going to skip for now because Sue's going to go into detail about it later. And then there's the idea of repeatability, selectivity, stability, and reproducible detection. So this set of guidelines, we're going to walk through just a little bit how you might set it up, and then we're going to look at some of the data from the vitamin D binding in a bit. So the first is the mini validation of repeatability. So at least in my lab, we affectionately call this the 5x5 or potentially the 3x3, depending on how lazy we're being. So the 5x5 idea is that you would take different concentrations of your analyte, so kind of like a mini calibration curve, and then you would measure it multiple times on multiple days, prepare it multiple times on multiple days, measure on multiple times, multiple days. If it is a good target, it should be very reproducible. You should be getting the same signal every time, even if you're preparing it on multiple days, even if you're measuring it on multiple days. And Andy showed us a little bit of that before when he was showing us like his big CV table. That was similar in that it was a stability assay. The key point with this is that it's measuring your intra and inter variability for each of your targets. So if your target is really hard to get the digestion reproducible, it would show up in this kind of an assay. It's a little bit of work, at least for me, because you've got to be, you got to have the time to prepare those samples on multiple days, and then you would analyze them all. So it takes a little bit of experimental planning for that. So the experiment 3 in the CPTAC is the selectivity. So you would have 6 different matrices. So I guess for my yeast, maybe I would have 6 different flasks and harvest my 6 different flasks and prepare my 6 different lysates to be 6 replicates of the same matrix. I would put my peptides a blank with none of my peptides. I would have 25 times the LOQ like I would determine with Sue, and then like 50 times the LOQ that I would determine with Sue. And the point with this experiment would be you would see maybe one of these biological matrices has some interference that I didn't happen to see in my calibration curve. So in yeast, maybe I don't have to worry about this quite as much, but in maybe like human, like biological human plasma, maybe between different people or different pools of people, different populations, I would have an interference that I didn't see in one of my other matrices. The fourth is stability, and this is the, I know Andy talked about this a little bit before. This is the one where maybe you would take your sample, you would, in one experiment, maybe just put it in your auto sampler and measure it different days. Is the peptide target degrading in the sample? Is it adhering to the plastic or something? And then you would also do a freeze thaw test. So you'd put all of your aliquots, you'd prep them all, you'd put all those aliquots in your freezer, and every day you would pull 3 aliquots. So after 6 hours, you would pull an aliquot. After 3 hours and a freeze thaw, and then another 3 hours, you would pull an aliquot. And then maybe you'd leave it for a whole month and pull it and analyze that. If the peptide, if anything weird is happening in the freezer, which we like to think it's -80, nothing weird's going to happen, but it does, modifications happen, you would be able to assess this in this experiment. So I think this one is maybe the most intuitive of all of them, and it's kind of like maybe the easiest to at least wrap my head around. Doing this, testing this at 4C is also a good idea. If you're auto sampler, you happen to like load all of your samples on Friday and then keep running it all weekend till Monday. Knowing that your peptide is not going to degrade over the weekend is good to know. And then finally, the reproducible detection. So I told you before about like the mini 3x3 or 3x5. This is doing the full 5x5 experiment, where you would have day 1 and you would prepare 5 samples. Day 2, prepare 5 samples, and then analyze the coefficients of variation for those, for the inter and intra reproducibility of your protocol. It's nice to do this with different sample handlers too, if you happen to have like a core facility. I've seen this done where different operators were doing each of the days and then analyzing the reproducibility of your peptide targets if they're being prepared on different days. So Andy showed this figure. Oh, one question in the front. It's just 5 by 5 instead of 3 by 5. 3 by 5 is nice to do at the beginning before you spend all the time doing all the other experiments, because you can very quickly eliminate a target potentially. Because the whole goal is kind of to eliminate targets. So if you started, for example, with just plugging into Skyline for one protein, give me all the possible peptides I could use as a candidate target, Skyline might say, cool, here's 46 options, or here's 48 options. You might be able to filter down this list based on peptide length, like we were talking about with my PMA1 protein and all the different peptides. Are there any trends? One trend might be peptide length. So you might be able to eliminate candidates by saying between 6 and 25 amino acids. You might be able to say, okay, I don't want any methionines, because those tend to be modified and have reproducibility problems. For that good precision, you might be able to do like a 3 by 5 or a 3 by 3 experiment and say, wow, well, some of these peptides degrade really quick, or some of these peptides don't digest reproducibility. You can do a freeze thaw to eliminate more candidates. You can look in different plasma matrices from different populations to say, oh, one of these peptides has transition interference that I shouldn't use. And then you should end up, at the end of the day, all of these calibration validation experiments should narrow down your list pretty quickly. You should eliminate a lot of targets, which is kind of sad to see at first, but you're being left with the best candidates. So next, we're going to look at the calibration validation experiments that were performed for the vitamin D binding work. Are there any questions before we start looking at the data? Yes? Control for LC variability. How do you control? Yeah. For targeted proteomics, I think the most common way of controlling for LC variation is doing those internal standards and doing peak areas. That should control for the variation in LCMS, because you're spiking in that heavy standard. In larger scale proteomics, we try to do computational methods, dynamic time retention alignment, but we don't do that typically in targeted proteomics. If your LC is so variable that it's significantly adding to irreproducibility, it's kind of nice if you capture it in these so that you know. I don't know if any additional things to say. Yeah, for the variability, yeah. Do you guys do anything additional? I don't think so. For LC variability. Yeah, I think internal standards are pretty much it. Yeah, we're going to be talking a lot about system suitability tomorrow, like basically all day. We're talking about system suitability and methods QC. I don't start running any samples until system suitability passes. All right.
Any suggestions on internal standards?
Oh, Sue's got a whole section on standards. So we're going to talk lots about those. Just for fun, I don't use any. So we'll also talk about that later, too.